welcome back to part 5 of our look at the immune system. And in this video, we are going to focus on T cells and how T cells become activated um, to target specific antigens. Remember, the idea about this is that this is adaptive or acquired immunity. And so the T cells, once they've passed the positive and negative selection test, go to the medulla to finalize their maturation, and then they are released into the lymph nodes and the bloodstream for circulation, so that um, they can go ahead and be specialized for a specific antigen. What causes that specialization, remember, is the interactions with the MHC1 and or MHC2 proteins. And so how does this happen? How do we get these T cells to become specific, antigen specific? And the answer to that is there's a three-step process. The first thing that has to happen is the T cells has to have recognition of the antigen. And so we have to confirm that the antigen itself is not of self, meaning that it is pathogenic, or that it is a mutation or an altered form of a antigen or a protein that is self. And so that's step one. That involves, as we know from the last video, that recognition part involves the MHC1 and or MHC2 proteins, which we're going to talk a little bit more about here in a couple seconds. Once there is confirmation that there is an antigen that does not belong, then the T cells have to react. And in order to react, the cytotoxic T cells, the killer T cells, they are directly going to respond to the MHC1 proteins, and the helper T cells are going to respond to the MH2 proteins, which will then initiate the helper T cells to go ahead and stimulate the cytotoxic T cells, or the killer T cells. Remember, MHC2 proteins are located on things like dendritic cells. They are located on things like B cells. They are located on things like macrophages. All right, and so there's a little bit of an intermediary step with that. And then the third step in this whole process is to remember. Right? We create these memory T cells that stores the interaction with the antigen, which allows the immune system to mount an immune response faster on the second exposure to that antigen. All right. So we're going to see on the next slide that there are actually there's two different pathways in which T cells can become active, and part of that is whether or not you're dealing with an MHC1 protein that is bringing the antigen, or whether or not you're bringing in or whether it's an MHC2 protein that is bringing the antigen. If we're going to take the CD4 pathway, if we're going to take the CD4 pathway, which I introduced in the last video, remember that CD4 stands for classification determinant. And so this is going to go ahead and determine the type of pathway that we are going to take. CD4, right, classification determinant 4 pathway cells, become helper T cells. Right, they become helper T cells. And they are restricted to only binding to MHC2 proteins. Right? And so they are your antigen presenting cells. They are your true antigen presenting cells, your macrophages, your dendritic cells, and your B cells. Um, Classification determinant 8 pathway cells, or CD8 cells, become your cytotoxic cells, your cytotoxic T cells. And they only interact with MHC1 proteins. And remember, they're the cells that are going to give that direct lethal hit. Um, and what they do is, and here's why this makes sense. MHC1 proteins are found on every single surface of every single cell except red blood cells. And you want your cytotoxic cells to be very aware of all of the cells that it comes in contact with to be able to identify that antigen. Whereas your MHC2, your androgen presenting cells, or your antigen presenting cells, your APCs, um, they're dealing with the helper T cells because pretty much we're saying, all right, yeah, this is an antigen that we've already come in contact with, and so let's go ahead and get this defense system up and working rather quickly. And so that is what you're seeing here. You're seeing that differentiation between the CD4 and the CD8 cell. And so over here, this is a CD4 over here, which is only going to talk to the APC cells, the dendritic cells, the macrophages, or the B cells. Whereas your CD8 cells over here, your CD8 cells are only going to communicate with MHC1 proteins. Now remember that APCs have both receptors. They have both MHC1 and MHC2 receptors. They are the only cell that has both MHC1 and MHC2 receptors. All right. And so you can see down here, and we're going to look at this in a little bit more detail. These cells down here are becoming your helper T cells, which we know what their role is going to be. We know that there's 3 different ones, helper T1, helper T2, and helper T17. And then over here, these guys here are going to become your cytotoxic cells over here. And so what does this look like? What does this look like? And so here, step 1, if you would, in this whole process, step 1 is the APC. The APC, whether it is dealing with an MHC2 or an MHC1, all right, we're going to assume that the APC cell is, we're going to say that this is what's bringing it. Right, remember, this could be just about anything. The APC is going to go ahead with the antigen bound, right, with the antigen bound, and by the way, the antigen itself becomes attached to this carrier receptor, which is called an epitope. It's called an epitope. And so that antigen is stuck to the epitope. The cytotoxic or the helper T cell, depending if we're dealing with an MHC1 or an MHC2, attaches itself to the epitope, and it confirms that the antigen is definitely not of self. All right. Then it needs to double check to make sure that this APC is not some kind of intruder. All right. And so all APCs have an additional protein that's called a co-stimulation protein. And that is what you are seeing right here. And that co-stimulation protein is simply a failsafe. It's a backup. And so what will happen is really in step 2, right, the cytotoxic or the helper T cell binds also to the co-stimulation protein. And it says, yep, you are a verified APC. The MHC1 or MHC2 receptor is legit. That epitope is legit with the with the antigen, and we can go ahead and then engage in step 3. Right? And during step 3, we have cloning of the cytotoxic cell or the helper T cell. All right. So remember, if we're dealing with CD4 or CD8, all right. So if we're dealing with if we're dealing with, I'm sorry, if we're dealing with CD4, all right. If we're dealing with CD4, remember that we are dealing with MHC2s. Right, we're dealing with MHC2s, and so that's going to create the helper T cells. And if we're dealing with CD8, then we're dealing with MHC1 proteins, and that is going to generate the cytotoxic. Right. So again, if we come back over here, right, these are your cytotoxic cells, and so these came from CD8, right, dealing with MHC1, all right, proteins. The helper T cells would have come from CD4 and MHC2 proteins. And then you also have some memory T cells that are going to go ahead and be created. Now, again, the difference here is cytotoxic T cells, or your TC cells, they're going to give a lethal hit. They're going to actually attach directly to the cell. Right? And when we talk about that lethal hit, remember we're dealing with perforin, and perforin is going to create a hole in the cell by which then those cytotoxic T cells can release granzymes to go ahead and lead to apoptosis. Whereas the helper T cells, they're going to stimulate B cells, they're going to stimulate neutrophils, they're going to stimulate macrophages, and they will stimulate more cytotoxic T cells. Right, so they will go ahead and stimulate more cytotoxic T cells. Um, Not only do your cytotoxic T cells have perforin and granzymes by which it can go ahead and force apoptosis, but they can also secrete something called TNF. Right? TNF is tumor necrosis factor. Tumor necrosis factor. And TNF is specific to cancer cells. Right? And so that is what the cytotoxic T cells would secrete in the presence of a cancerous cell. It would be TNF, tumor necrosis factor. And that does the same thing that the perforin and the granzymes would do. It forces that cell into apoptosis. All right. And so this here is, again, just whether or not it is a MHC1 or an MHC2 protein that we are dealing with. And so that is what you're seeing in this diagram here. Interestingly enough, what you're seeing right here is the helper T cell is binding to the MHC2. It's going to force the CD4 pathway. Right? And then down here, the cytotoxic is the CD8 with the MHC1 proteins. But what's being secreted right here, and this is not well described in the diagram, at least not obviously, but the helper T cell is secreting interleukins, and that's interleukins right there. And so you're seeing interleukins that are also helping to stimulate the replication of cytotoxic cells. And so these guys right here that's being secreted by the helper T cells. And so again, this here is just another overview of that process. Right? And down at the bottom, you're seeing your cytotoxic cells, which are directly attached to the enemy cell. And you're seeing the helper T cell that is now working with that APC cell to go ahead and stimulate more response. And so we're going to go ahead and stop here. I know this is a shorter video for you guys. We're going to stop here. We're going to kind of let you rewind this and kind of digest this. And when we come back, we will look at humoral immunity or antibody mediated immunity. Hopefully this is all starting to make sense to you. Again, jot down those questions. Let me know what you need. And hey, I'll catch you on the flip side.